Hello everyone, I am at Scottsdale College right now. I am currently looking for all of the creators of Brittany's Movie. It's supposed to be really good, coming out in theaters soon. So I gotta look somewhere and find out where they are and interview them a little bit, kind of find out something about them. Oh my god, there they are. Let's go find them. Okay. They're right over there. Trip! There they are. Oh god. Oh my god. I'm like your guys' biggest fan. Oh god. What's your name? Okay, so what inspired you to make the movie? Sounds interesting. You guys single?
Maybe.
Signing off. Bye.
The action potential starts in the axon and goes down, and as it goes down, it opens calcium channels on the sides. The calcium forces the synaptic vesicles down, they fuse with the membrane, and they release their acetylcholine into the synaptic cleft. The acetylcholine attaches to the acetylcholine receptors and opens other channels in the membrane, allowing sodium to pass into the membrane. Sodium causes depolarization in the muscle, which travels down the muscle as an electrical signal. Then an enzyme in the muscle called acetylcholinesterase is released back into the synaptic cleft to disintegrate and break apart the acetylcholine so that the contraction stops. Then potassium is released into the cleft so that, Then potassium is released back into the synaptic cleft, which causes polarization. This is the whole muscle. Inside each whole muscle are fascicles, which are the orange ones. Inside each fascicle are muscle fibers, which inside of them are myofibrils. Then calcium is pumped back into the sarcoplasmic reticulum and attached to calsequestrin, which is a molecule that holds calcium. And this allows tropomyosin to again cover the active site, which results in the relaxation of the muscle. The AP travels down the sarcolemma into the T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum into the sarcoplasm of the muscle cell. The whole thing is a sarcomere. The Z lines are on the end. The M line goes down the middle. From thin filament to the Z line is called the I band. From one side of a thick filament to the other is called the A band. And then the middle is called the H band. Calcium floods over the thin filaments and binds to the troponin, which pulls the tropomyosin off the active site on the actin. The high-energy myosin heads leap to bind to the active sites on the thin filaments, and this forms a cross bridge. Then the myosin head pivots to the low-energy state, which releases ADP and moves the thin filaments over the thick filaments, which is called a power stroke. Then the ATP binds to the myosin head, which causes it to break free from the active site on the actin. Then the myosin head again becomes energized as ATP and converts back to the ADP. Then calcium,